We are going to go over genetics questions. The first one here. A somatic cell always has dash of chromosomes. We'll remove what's not true. Pairs of chromosomes. All cells, we know that they've got pairs of chromosomes, whether they are somatic cells or sex cells. And somatic cells don't have two pairs of chromosomes. And we can't say haploid number. Haploid is in sex cells. When you're talking about somatic, these are body cells, they've got a diploid number. And we can't say 23 pairs because there are only 23 pairs when it is in human beings and not in any other somatic cell. What if we are talking about a somatic cell for a plant or just any other animal? Question number 52. The primary growth phase of a cell is there. So when we're talking about the cell cycle, it's divided into the M phase and the interphase. The interphase has got three parts. There is the G1, which is the first growth or gap 1 phase. There is the S phase, and then there is the G2 phase. Okay, so this is the one which is different. The rest, which is not shaded, is the interphase. So G1, this is where the first growth of the cell organelles get to happen and the cell organelles gets to grow. In the S phase, this is where we've got replication of DNA. And in the G2 phase, there is a continuation of the growth of the cell and making sure that the cell is ready for division. So the first growth is in G1. 53. Choose the statement which is true about G2 phase. The phase in which DNA replicates. DNA replicates during the S phase and not the G2 phase. The phase in which the cell grows before DNA replication. Before DNA replication, replication is in the S phase. So before it is in the G1. So G2 is after. Therefore, the phase after DNA replication, this is true because it is after the S phase. The same phase as prophase? No. Prophase is found in the mitotic or meiosis phase. So the answer there is option number 3. Interphase in the cell cycle is the time interval between meiosis 1 and meiosis 2. This is not true. Meiosis 1 and meiosis 2 is found just in the meiotic phase. Composed of G1, G2, M phase, and S. Because of M, this will no longer be interphase because interphase is only G1, S, and G2. M is another phase. The stage when chromosomes are visible. No. Interphase, the chromosomes are in their chromatin state. So they have to be condensed in prophase, that is in the mitotic phase. And for the time of cell growth and duplication, this is true. The division of the cell is not true because this is during either meiosis or mitosis. Okay, let's consider question number 55. Crossing over is identified by. Now, when I'm talking about crossing over, you have got first this. This is a chromosome. So when this chromosome makes a copy of itself, it's going to have a sister. So now they are going to be called sister chromatids. They fully contain the same information. If this one is talking about blue color here, even this one is going to talk about blue color here. Okay, now remember chromosomes are supposed to be homologous. So we're going to have an homologous pair like that. So even this homologous pair is going to make a copy of itself in that man. So if this one is talking about blue color at this point, this one will also be talking about color, but not the same color. This will maybe be talking about white color, but they are talking about the same thing at the same position. They are all talking about color. If at this position, this one is saying you need brown eyes, even at this position it will be brown eyes because they're what? They are all the same. They are sisters. They are just copies of each other. But this one will be talking also about eye color, but it might be talking about blue or something, a different color from the other one. Even this one will be talking about the same thing that this other one is talking about. So these two are called homologous, and then these two are called sisters of each other, sister chromatids. And so when crossing over happens, let me just get to put it like that. So the coming together of these two chromosomes is called synapsis, and this is going to happen during prophase 1, and now there will be crossing over that gets to happen. So it is during the process of crossing over that there is also genetic recombination in the cell, and we're going to have something like this. So let me again just get to shade this so we see how they get to differ, those which we have at the beginning and those we're going to get at the end. So this point when these get to meet is called the chiasma, and these which are getting to cross over are non-sister chromatids. We can see this chromatid is coming from one chromosome, this other one is coming from also another pair of homologous chromosome. Actually these two are a pair, they are called sisters of each other, they are the same. Okay, so homologous chromosomes are identified by the same centromere, the same location of genes, and the same genes that we're talking about. If this gene is about eye color, even this other one will be talking about eye color from the other chromosome if this one is also talking about eye color. So after crossing over gets to happen, what you are going to see is these are going to now be recombinant. Okay, so what you're going to see is because there was crossing over, there will be a part like that which is going to go from one chromosome which is shaded, and then it will be on another one, and also this one which was shaded, everything they've exchanged positions. This position has exchanged with this one, but these two are still homologous. Why? Because they still contain the same position of the centromere, and they still contain the same information. It's just that this one here was talking about the color of the eyes, blue. This one was talking about the color of the eyes, let's say brown. But now since they've swapped, this position is talking about blue color, now this one is talking about brown color. These two are the ones which have just changed. They were supposed to be talking about the same color since they are the sisters of each other. In other words, they are just copies of each other. But because of recombination, they are no longer talking about the same thing. But it is still color, but not the same color. So they are still homologous. Okay. So now how do you get to identify crossing over? Crossing over is identified by recombinant homologous chromosomes. So this won't be true. The recombinant homologous chromosomes do not identify crossing over. Crossing over is just a point where these chromosomes get to intertwine. Just that point where they get to intertwine. That is the crossing over. That's how you can identify this point where they are intertwining. Okay. Of course, this is another one like that, and also another part like that. So that's the point where they're intertwined. Now, chiasmata between sister chromatids. This point where they get to intertwine, these are not sisters of each other. So their chiasmata is not formed between two sister chromatids, but between non-sisters. So recombinant non-homologous chromosome. When you're talking about the recombinant, that is after crossing over. So we are talking about crossing over itself. And then chiasma between non-sister chromatids, this is true. So the chiasma is formed between two non-sister chromatids, and whenever we see that, then that is crossing over. Okay, now let's go to 56. Choose the statement which is true about the human reproductive cycle. The mother cell from which gametes are produced are diploid. This is true. Why? Because when we're talking about the process by which gametes are formed, this is a process of meiosis. And we've got meiosis 1. By the end of meiosis 1, during telophase 1, what you are going to have is now from 2n, which is the diploid number of cells, to n. So it is during telophase 1 that the chromosomes are no longer diploid but haploid. Okay. Meiosis is important because it is a source of genetic variation. This is also true. The same recombination that happens during prophase 1 makes sure that there is genetic variation. The male reproductive organs are called testes. This is also true. So this is telling us that all of the above statements are true. So that would be the best option then. Let's consider 57. Which of the following features of cell division are very different for animals and plants? Now, prophase, both in animals and plants, is the same. When we're talking about mitosis, it's the same. Metaphase is also the same. Anaphase is the same. Telophase is the same. Now, when we're talking about cytokinesis, it's different from plants and animals in that for plants, for the cytoplasm to divide is going to form what is called the cell wall plate. Okay, that's a cell wall plate or just a cell plate. And when you're talking about animals, in animals there will be the cleavage furrow. That is what differentiates cytokinesis in plants and animals, the division of the cytoplasm after telophase. Now 58. During dash, synapsis occurs in the phase called dash. Well, we know that synapsis happens in meiosis, and that is during the prophase 1. So the best option we can give here is option 2, meiosis. During meiosis, synapsis occurs in the phase called prophase. 59. Choose the correct statement. The cell plate is formed in animal cells during cytokinesis. Now, cell plate is not formed in animal cells, it is formed in plant cells. Chromosomes align themselves along the equatorial plane during anaphase. No. The alignment of chromosomes on the equatorial plate is during metaphase. Centrioles are present in both animals and plants. This is true. It is from the centrioles that we get the spindle fibers which get to attach to the kinetochores during the process of self-division. So I can get to illustrate this where we've got this chromosome here and this other chromosome. Well, this is one chromosome where I've got sister chromatids. Now this is the centriole, this another centriole here. So the only way that the division of this can happen is when you have got the centrioles from where the spindle fibers are going to come and attach here so that they can be able to pull these sister chromatids away from each other. Now let's look at the other option. DNA replicates during the M phase. That's not true. Replication of DNA happens during the S phase. Question 60. Choose the genetic term which matches with the corresponding description. Haploid, any chromosome other than the sex chromosome. This is not true. Any chromosome other than the sex chromosome is called an autosome. Any cell other than a sex cell is called a somatic cell. So we need to get to know that. Meiosis is called reduction division. This is true. That is because when you've started with a diploid number, you're going to end up with a haploid number. Cytokinesis, number of chromosomes in the gamete. This is not true. Number of chromosomes in the gamete is called haploid. Okay, half the number of chromosomes. And autosomes site on the chromosome to which spindle fibers are attached. This is not true. This is the centromere. That is the site where spindle fibers get to attach. And centromere, division of the cytoplasm. This is not also true. Division of the cytoplasm is called cytokinesis. So the best option is option number 2. 61. Dash most closely resembles events of mitosis except that the cells are dash. Now, when you're talking about mitosis, mitosis starts with prophase. Of course, the first stage is interphase where the cell gets to replicate. But when you're talking about prophase, this is where the nucleolus is going to disappear. The nuclear membrane is also going to disappear. Okay, that is during, and the chromatin gets to decondense or condense into chromatid. That is during prophase. And after prophase, we can remember this as IPMAT. Interphase is the very first. After prophase, we've got metaphase. Now, the condensed chromosomes get to align on the equator. And here, the centrioles have moved to the opposite sides. They get to form spindle fibers which get to attach to the center of the chromosomes, which is the centromere. And the proteins which can hold the spindle fibers Those are called kinetocos or kinetoc. So this is the metaphase. And after the metaphase, what we are going to see is the sister chromatids are going to be pulled away from each other. They'll be away from the center, and that is now the anaphase, meaning away. And the last one was just going to be telophase, where these almost gets to split. You are going to have more like that in telophase, and everything that disappeared during prophase is going to reappear in telophase. Now the difference with meiosis is, when you are talking about meiosis divided into 1 and 2, for meiosis 1 you've got first prophase 1. Prophase 1 has to do with the genetic recombination, the disappearance of the nucleolus, and the disappearance of the nuclear membrane. And this is where we're going to have also genetic recombination, because they'll be crossing over. But the difference again is we're going to have not only chromosomes like we have during the prophase here, only one like that, one chromosome. We are going to have homologous chromosomes which have made a copy of themselves. After prophase 1, we are going to have metaphase 1. So during metaphase 1, it's the homologous chromosomes which are going to align the equator. And the way they get to align is different the way they get to align in metaphase of mitosis. In that here, the homologous chromosomes get to align like that. And during the first metaphase, what you are splitting is the homologous chromosome. So they are going to align like that on the equator here. And you are going to split them, that is during anaphase 1. So anaphase 1 is going to split this sister, the homologous chromosomes. So we're going to have one here, one at the end, again one here, one at the very end. And now this is anaphase 1. The last one will be now telophase 1. Telophase 1 makes sure that the diploid number of chromosomes which is started with is reduced to a haploid number. So you're only going to have half of what you had at the beginning. Now after telophase 1, you're going to step into telophase 2. Meiosis 2. Starting again with prophase 2. Now the thing is, in prophase 2, we don't have any. There is nothing like genetic recombination again. Genetic recombination already happened. So during anaphase 1, you have split. The only difference is this: we don't have genetic recombination in prophase 2. It's all the same, just like prophase of mitosis. And here, even metaphase 2 is just the same like metaphase of mitosis, because only one chromosome is going to align at the center like that. Okay. And also the anaphase 2. Anaphase 2 is the one which is going to now separate these sister chromatids. Anaphase 1 separated homologous chromosomes, but anaphase 2 is going to separate sister chromatids. So anaphase 2 is the same as mitosis. Let me not say anaphase 2, but meiosis 2 is the same as mitosis. The only difference is that for mitosis you talk about a diploid number, but for meiosis 2, remember telophase 1 reduced the diploid number to haploid. So for meiosis 2 you are talking of a haploid number, but all in all they are all the same. So thank you so much. We'll end here for this one.